All right. To kick off our cellular metabolism unit, we're going to be looking at how chemistry and biology are related to each other. We're going to be looking at metabolism and energy and enzymes. So we're going to be combining two different sciences so that we can better understand cellular and respiration and photosynthesis when we get to them. So we're going to start off talking about kind of a fun enzyme, lactase. So as many of you are aware, not all that long ago in human history, it was, there weren't a lot of people that could digest the sugar found in milk, which is lactose. We still have lactose intolerance today, but some of us still produce the enzyme that breaks it down into adulthood, which is actually a change in the human population over the past 10,000 years. So babies, of course, could digest milk, but once they got past that infancy stage when they weren't reliant on it anymore, they would lose the ability. So we're going to be looking at the cellular metabolism that allows for that to work, and then just enzymes in general. So first, we'll talk about energy. Energy, if you remember from physical science, is the ability to do work. And there were two main types of energy. You had kinetic and you had potential. So kinetic was energy of motion, chemical was stored energy. So one of the big biology ideas here is that biological systems use free energy and molecular building blocks to grow, to reproduce, and to maintain dynamic homeostasis. And so we are going to be talking about how it's maintaining its dynamic homeostasis. So the first thing we have to talk about is the laws of thermodynamics, just a refresher on those. The first law of thermodynamics is the law of conservation of energy. So energy can neither be created nor destroyed. The second law of thermodynamics is that energy cannot be changed without a loss of usable energy, so usually due to heat. And the laws of thermodynamics as they apply to biology, so if you think about it, living organisms must obtain energy from their surroundings, so we eat it, and transform it into usable energy to do work. So we have to take whatever food we ate or whatever drink we ingest and use it for energy. Energy is obtained from food sources, the sun, chemicals, and the transfer of energy. And during that transfer of energy, the energy of changing my iced tea here into usable work for myself, I'm going to lose some energy to heat. All right, so enzymes are the biological catalysts of our cells, and they each have, there's tons of enzymes, they have different functions. The first one that we're going to talk about is lactase. Lactase is the enzyme that's responsible for breaking down that lactose that you would take in milk, and that is in the small intestine. You have amylase, which is starch to simple sugar, so you'll find that in your saliva. Pepsin is the one that takes proteins and breaks them down into amino acids. That's in your stomach. And lipase, which so it has that fat. So it is fats to fatty acids, and that is going to be in your pancreas and intestine. Then you have alcohol dehydrogenase, and that's alcohol into non-toxic substances and sugar, and that is found in your liver and stomach. So those are some enzymes that are worth noting. Biology, it's like, so how do you spot an enzyme in your everyday life? So each enzyme is specific for a substrate, and most enzyme names end in the letters ASE or A's. So notice when I went over those enzymes, I said lactase, amylase, lipase, dehydrogenase, usually end in that A's. So the enzyme that breaks down sucrose, which is sugar, is sucrase. So it's just changing that ending to ASE instead of the OSE. So then the enzyme that breaks down lactose is lactase, changing it to ASE from OSE. So the sugar lactose consists of a galactose and a glucose molecule joined by a glycosidic bond. So basically these are two hexagonal chemical structures that are going to be connected to each other. And these simple sugars can be absorbed by the cells of your small intestine. Lactase lowers the energy needed to actually break that bond. So lactose is those two sugars together. The enzyme makes it easier for the bond to break, so then they form into their respective glucose and galactose molecules. And that's what you want to have enter your cells. So lactase helps with that. So if you remember anything about, your regular biology class, enzymes are proteins. So they are one of the macromolecules that's responsible. It's one of the building blocks of life. Proteins, your muscles are made of proteins. You function on proteins. Without proteins, we all would not exist. So proteins can be formed several different ways. Their building blocks are amino acids, and they're joined together by either by dehydration synthesis, so the removing of a water, so the amino acids can come together and make a protein. Hydration or hydrolysis is actually you add water and you break apart the amino acids. So you can combine them by removing water and break them apart by adding water. Lactase is made up of 4 subunits. Each has 1,023 amino acids. So 4 subunits, 1,023 amino acids each, which gives you a total of 4,092. So, kind of gives you the idea of the structure. Now let's look at how enzymes work so we can apply it to lactase. So an enzyme is a protein molecule that functions as an organic catalyst. So catalyst means that it speeds up reactions. A substrate, reactants in the enzymatic reaction. So this is what the enzyme attaches to. So it's taking the reactants and it's attaching to them. So if you think about chemical equations, how you have reactants and products, we're looking at the reactant side of that. So then you have to have the energy of activation, and that is the energy required to cause the reaction to occur. So there has to be a set amount of energy there for the reaction to occur. So the enzyme will bind to a substrate, and then the activation energy is going to be what gets the reaction going. All right, so properties of enzymes, once again, made of protein to speed up reactions, remain unchanged after each reaction. So if you remember or took a chemistry class, you know that when you go through the chemical reaction, the enzyme or the catalyst is not used up in the chemical reaction. So an experiment we would do if we were meeting in class as normal would be to see how well yeast digests different sugars, but we won't quite be able to do that one. So just continue talking about enzymes and we'll get back to the whole lactase thing hopefully at the end. I only have about 2 minutes left before the video gets too long though. So factors affecting enzymatic speed, so substrate concentration, how much of the substrate is present, the temperature, the pH, so the acidity, or whether it's acidic or basic, and then enzyme concentration, how many enzymes there are. So keep in mind that enzymes can be denatured if they're in a high temp or a pH that they can't exist in, so they will be no longer effective, so they're not going to speed up that particular chemical reaction. So enzymes are responsible for lowering the activation energy of a chemical reaction, and that's going to lead us to exergonic and endergonic reactions, which I will talk about in the next short video.